So what this is going to do is that gene list that I put in with all that fold change and correlation information, it's going to compare my list to all the 20,000, the list from all the 20,000 different studies that were out there. I don't know how many lists are in there, but there's a lot. And as we do this, what we would expect, obviously, is some breast cancer grade classification study. So what it's doing is it's pulling out data sets that I didn't use in the original one. Maybe I use this data set next time and combine it to the analysis. So you're almost using this stuff like bait, right? I got my bait, I got a really kick butt, breast cancer gene list, I throw it in there, and now I'm fishing out other things that are similar to it. I do this a lot with a lot of different types of cancers. Is what I found is I see a lot of lung cancer that looks more like bladder cancer than it does like other lung cancers. And this will allow you to kind of see what is your cancer, what is your condition most like? And a lot of times it'll surprise the heck out of you. That when I throw this stuff in, I see a lot of immune stuff, which you wouldn't expect from cancer. But when you start thinking about it, you're like, oh yeah, of course. The cancer has to somehow hide from the immune system, so there's got to be something going on with the immune system. So I'll pick that up, and you can actually just take those genes and actually look at those. Okay, so again, curated studies. This allows us to, we can add other things to our meta-analysis if we wanted to. Oh wow, this is really close like that. Yeah. What it'll do is, and then if I click on this, it's telling me why is it bringing up this study? So in my list, I had 969 in my gene list. 572 of them were upregulated and 397 were downregulated. In this list, this is breast cancer, histology grade 3 versus 1. There they had this many lists. So they had 4,979 genes that were differentially expressed. My gene list is getting grouped with theirs because when I combine them, out of my gene list, all of them except 3 were found in the other data set. Not only that, but when I look at whether those genes go up and down and mine go up and down in the same direction, when we look at this, basically almost all of them have the same directionality. They're going up or down with the severity of the grade of the cancer. Here, there's only like 2 genes that do something different between the data sets. Given this fact, that's why we're getting a p-value of 1 times 10 to the negative 323rd. And by the way, I did talk to the Illumina people. Like I was at one of their seminars and I was talking to their science people. Like you can't get a p-value of negative 323. They can. He spent about 15 minutes, but he convinced me that this is possible. They have some pretty big computers there, but okay, let me close out of this. Let me show you some other things. Absolutely.
Go back down there for a second. So you compared grade 3 to grade 1 according to the text there, right?
Well, that's the list I'm comparing to.
Right, but so when you look at overexpressed genes, it would be overexpressed in a grade 3 cancer versus a grade 1 cancer. Is that right? I'm just trying to
grade 3 so I took grade 3 either versus grade 2 or grade 1.
Yeah, I'm just trying to figure out what's over and what's under.
So the way Illumina sets it up is that they always put the more severe form or the disease versus the control. So it's kind of convenient that way. And you do see that sometimes you'll see something flipped and it's kind of a hard comparison. But in this case, ours was a kind of combination of stuff. Grade 3 versus grade 1 or grade 2. Here in this gene list that they're looking at, it's just grade 3 versus grade 1 in this comparison. Does that make sense?
So you could have put them in either order and it would be comparing essentially grade 1 is your control, grade 3 is your bad condition.
Right, and if I flipped it and say in my list I had grade 1 versus grade 3, instead of saying that this was a positive correlation, so you see right here this correlation direction, you see that positive? That means it's going in the same direction. If I would have flipped it, then it would have been negative. And that's what you see. So not only are you basically finding things that are similar to your gene list, but you're also finding things that are completely dissimilar. And that's important as well. And you kind of see this as we go here. Any other questions up until this point? I promise I'll only go for another 15 minutes. Everybody's like, so much information. Turkey sandwich is so good. Okay.
About differentiating between human and mouse. So if you're growing tumors, a human tumor in a mouse, can you differentiate the genes of the mouse versus the humans?
With this tool?
Yes.
You would have to do that beforehand before you uploaded it. Okay. Or you could just basically, I mean, that's an RNA-seq study, right?
Yep.
I mean, you're going to have sequence on that. If you could just upload your human data genes that you wanted, once you've made that determination, you could definitely do that. And I have done that. And so there's a lot of xenograph kind of studies on here as well. So yeah, that's absolutely doable. Okay, so let's see, curated studies, go to Body Atlas. I kind of showed you that. I'm running out of time. We can go to, here's the cool one. I can go to Pharmaco Atlas, right? I'm gonna click on that. Any MDs in the room? No MDs? MDs love the drugs. So when I start talking about the drugs, you just see like people, huh, what? Drugs, what? I can prescribe those. Here is what this does is it's bringing up all the chemicals that it knows, all of its studies where a drug was used that has similar effects to the gene list, the directionality of the gene list that we put in, right? Let me rank them. It's me rank them. So if we look at this top one, this chemical, diadzine, I'm so terrible with pronouncing stuff. I think we all have the same abilities. Like, you know, for me to understand higher math, I just kind of gave that, I'm going to give up pronouncing words. Has anybody heard of this chemical before? No? If you click on it, and you can click on the PubMed, and you can Google it. You can Google it right now. Somebody Google it. Or is anybody on their computer? No? All right. Don't Google it. It's okay. I'll tell you what it is. It's soy. It's a derivative of soy. What does soy turn into? Yes, science. And it's having it has a positive correlation to our genes. So if in these studies, and you can click here and find those studies where it's showing that, when they add this chemical, it makes their genes look like my genes. Or I should say my genes look like their genes. Does everybody understand that? Now obviously we don't want to cause cancer, right? So we're not looking at things with a positive correlation. Now we can look at this Pharmaco stuff and say, find me stuff that a negative correlation. Amphotericin. Anybody heard of that before? No? It is, so this is a thing that has a negative correlation, so this would be something maybe preventative. This is a derivative of vitamin D too. Anybody heard about cancer and vitamin D? Yeah, vitamin D is great for cancer. Like, people are using it a lot. We are seeing this now. We have now found a chemical that basically we could potentially use as the therapy, right? This is where this stuff goes, and this is where we're getting to. It's not that one little bit of piece of information that you find in your data, like that one gene. It's what do all those genes say that gets me to this point, right? I want big picture stuff. I want stuff that I can add to it. There's one more here I just wanted to see. Does anyone know what bisphenol A is? What is it? BPA. Does anyone know what BPA does? Mimics estrogen, right? Totally. What I want to show you, and I hope this kind of gets in your head, and you could do this for all these chemicals. I mean, it's awesome, actually. Is what you're trying to do is use all this, you're trying to build big pictures, right? This chemical page is a big picture. That these are things that affect lots of things in my system, and that's where I want to be. I don't want to be at the gene to gene level. I want to be up above saying, what can I do to influence the most things in my system? Let me do one more thing here, and then I'll do two more things. You can also do knockdown atlas. What this will do is it has all these different knockdown studies deposited into Illumina. And it knows that when it knocks this gene out here, what is this? Indian hedgehog, right? When you knock this gene out, it has a negative correlation to my genes. So what that would be telling me is that that's probably an oncogene, right? If I knock it out, it seems to turn my genes kind of the opposite, right? So that's probably an oncogene. If I knock something out and it has a positive correlation, that's probably my tumor suppressor. But what you can see is that what we're using is we took a bunch of studies, 24 studies. We found the middle of all those studies to get a really kick butt breast cancer gene list. We throw it in here, gene expression study. Now we get to the point where these might be genes that might be mutated, right? I went from mRNA to mutation. I can also go back down, and I'm going to show you. And you can download this list. I did a bunch of stuff with it. It's a pretty cool list, actually. Go to pathway enrichment. And I think this is what most people are familiar with, right? Pathways. Everybody loves pathways. I don't like pathways. I think most of these pathways were done 10, 15 years ago and in all kinds of different organisms and different studies. And I think it's a very simplistic, potentially out-of-date view to look at how this stuff goes. But we can, this will definitely measure this stuff. And if we look here, I'm looking for things overrepresented that are associated with breast cancer. And if you look, we can see those things. Cell cycle, mitosis. But as you'll see, what we can do is we can now select, I only want to see those things that have microarray targets, microRNA targets. So based on a just list of genes, we have a very good idea what microRNAs, what genes microRNAs inhibit. And that if we can look at the RNAs itself and find the pattern there, we can figure out what microRNAs are potentially altered. Does that make sense? Kind of killed that there. But now what I can say is I could say, okay, find me things with overrepresented targets of microRNAs. So now I'm saying, well, based on our gene list, I think microRNA 496 is down because all of these genes that it's known to inhibit seem to be going down for the most part. Yes. MicroRNA 200B. By the way, I started looking up a bunch of these, you could put in the microRNA and then keyword cancer. Yeah, they're all big targets. Again, I could do this for blood. Say I wanted to find a marker to, if we really wanted to get to, potentially find a cure for breast cancer or making a huge impact, early detection would be great, right? In something like a blood tissue. I can use this information. I've actually looked. There's a lot of a lot of studies out there looking at breast cancer and looking for biomarkers in blood. And sure enough, you can do the same in meta-analysis. And each one of those studies, I kind of looked at them, aren't that great. But when you combine them all, the final microRNA list is, I would totally test it in people, just to see what would happen for sure.